Okay. Hi, everybody. Happy to be here. I'm going to be presenting, as I said, this very broad topic, but I think you'll get to know why in a minute because I want to present just one piece of work, but give you some context about the broader questions that I'm interested in and the wider applicability of this approach than the specific problem that I'm going to be studying. And just to say that I'm a postdoc currently at the University of California in San Diego, and this is where you can find me. So we can look into that later. Okay, to start, let's just take a step back, right? The central goal of ecology and evolution is to try to understand the diversity of life on Earth. And generally, what are the patterns that we observe? So, what is it that we observe in ecology and evolution, right? If we just take a step back and really go back to basics. Well, in ecology, what we are interested in is the abundance and distribution of organisms. So, how many organisms are there at a given time in a given spatial location, right? And in evolution, what we generally look at as our pattern is the frequency and distribution of genes. So what is the proportion of a type, say this red type in this entire population of types, and how this changes with space and time. And generally, we are interested in dynamics, as I said, change in space and time. And so to kind of go back one step further, we ask ourselves, what is it that's actually happening that changes the abundance and distribution of organisms and the frequency and distribution of genes? And to do this, let's outline some of the processes and mechanisms in ecology and evolution, right? So how can a population change at a given spatial location? Well, organisms produce copies, so there's birth. They may die, so there's death. There's immigration and emigration, which I've summed up by dispersal. So this is kind of the classic growth equation. These are the kind of set of possibilities, and that's the only way a population can change, basically. And what are the kind of mechanisms that lead these populations to change? Well, it's their interactions with uh, the abiotic environment. So if you, for example, imagine a super stressful abiotic environment, some kind of weird temperature, for example, then this would obviously impact how organisms survive and reproduce in these environments. And the other type of mechanism is biotic interactions. So you can imagine if there's an organism which has a predator, then this directly affects its death rate. So these biotic and abiotic interactions impact the processes in ecology and evolution, in ecology specifically right now. And then let's move on to evolution, right? So what changes the frequency and distribution of genes? So if I see the change in frequency and distribution of genes, I can attribute it to a few different things. So the first one very classically is natural selection. When you have these different types indicated in different colors, these types have, and assuming that these types or this variation, they have here with G for genotypes, assuming that they produce differential number of copies, this would change the frequency and distribution of genes in the next time step. So this is kind of what we call natural selection. So and then there's obviously variation that can be brought in and taken out because of movement of these genes. And finally, there's mutation. So you have the creation of novel genetic variation. Also, there's stochasticity in all of these processes. So this is known as genetic drift, um, which also can change the frequency and distribution of genes. Okay, so now that we've talked about these two in separation, in fact, we can kind of notice that these are actually not as different as I made them out to be initially, even though that's how they're classically studied. But in fact, if we look at ecology and evolution, they're basically the same thing, but observed at different scales and trying to answer maybe slightly different questions. And this is not just like a very vague statement. There's actually something that comes about from these processes and mechanisms in both ecology and evolution, right? Uh, and to kind of outline that a little bit, if we consider these genes, right? So look at this gene right here. These give rise to phenotypes. right? So these are certain features of these organisms that you can sort of measure. And these phenotypes are what determine the biotic and abiotic interactions, right? So for example, you can imagine that a certain phenotype implies that you're better at dealing with a certain temperature. So, and this is kind of genetically encoded, which is why ecology influences evolution. So when you have a change in your frequency and distribution of genes, this would also change the abundance and distribution of organisms for this reason. And the second thing to notice is obviously that these birth, death, and dispersal processes are actually what give rise to the evolutionary mechanisms of natural selection, genetic drift, and gene flow. So uh, it's easy to see why, because when you have differential reproduction, you can just see this right here. Um, this is just your birth and death, differential survivorship and reproduction, dispersal, gene flow, genetic drift, stochasticity in all of these processes, and so on and so forth. So ecology and evolution are not just coupled at the level of in just like this vague way, but actually at the level of processes and mechanisms. And the other thing to note here is that these abiotic and biotic interactions, so this abiotic and biotic context of organisms actually directly influences selection, because this is what determines the birth, death, and dispersal rate. So this is kind of like the big picture of ecology and evolution put together. And so one thing that becomes very apparent when you think of this problem, right? This problem goes all the way up from genes to individuals to communities to ecosystems to and so on and so forth, right? And this is known as hierarchical organization. This is something that we learn very early on in biology. I don't know whether everybody here studied biology, but at least if you learn biology, this is one of the first properties of biological systems that you understand, that we learn, right? And this is that biological systems have this kind of hierarchical organization going from genes, which produce phenotypes, or individuals, which produce populations. So this is like a collection of individuals, communities, collection of species, ecosystem, collection of species, along with the abiotic environment. And this poses a challenge to studying ecology and evolution, because for two reasons, right? Two specific reasons. So the first reason is that when you go from genes to individuals, for example, the properties that you look at are completely different, right? So these are known as collective or aggregate properties. So if I go from individuals to populations, for example, right, individuals are what have, when I talk about individuals, I would talk about their birth, death, immigration, emigration rates, their phenotypes, and so on and so forth. But when I start talking about collection of individuals, now I start talking about population densities. When I start talking about collections of individuals of different species, I can talk about different metrics about species, right? The number of species, their diversity, and so on and so forth. So each time we go up this level of organization, there are new properties, that new aggregate properties, but there's also sometimes surprising behaviors which you would not expect based on these lower level properties. Sometimes this is known as emergence. This is the exact definition is a debate in the field, but this is one way in which it's defined. And not only is there hierarchical organization, but if we focus on one of these levels of organization, so let's say we focus at the level of these communities, right? There are complex interactions and structures. So what do I mean by this? So if you have multiple species in a community, it's not like all species are going to interact equally with all other species, right? You could have a situation where there's one species that is the predator of another species, but this species does not interact with another predator or with another prey in the same way or with the same strength. Like it does, for some reason, chooses not to interact with something, right? Or not even chooses, just like happens to is the right word, happens to not interact with something else. And this is kind of the case at every single level of organization. And finally, another property of ecological and evolutionary systems that poses a challenge to studying ecology, evolution, and ecoevolution together is that of spatial and temporal heterogeneity. So this means that all of these things, right, genes, individuals, populations, ecosystems, they are not the same in space and time, and there are spatial contingencies. So this means that there's a historic dependence on what just happened to be there for whatever reason. But also there are a lot of questions of spatial scale. So if you observe something at a smaller spatial scale versus a larger spatial scale, or temporal scales, right? If you look at very short-term evolution versus long-term evolution, short-term dynamics versus long-term dynamics. So there are these questions of spatial and temporal heterogeneity. And so when one wants to study this from a modeling perspective, one obviously has to do something which all modelers do, and this is a good thing. I want to emphasize again, this is a good thing, is that we make assumptions, right? So we make certain assumptions about the systems that we are studying so that we can study them more easily. And so when it comes to hierarchical organization, I'm just going to start giving some examples of simplifying assumptions, or just like outline what we usually assume. So when we study ecology and evolution, generally, people spend their entire careers sometimes on certain parts of this hierarchy of organization. And so one of the assumptions that people make, but not everybody makes this, right? This is like, I'm not trying to say that everybody is making these assumptions, but like often we make these assumptions where we study the one level of interest above or one level of interest below, but we are just right at the level of interest that that we want to study. So for example, if you want to study populations, we might model explicitly individuals and not model their genes, and this would probably then eliminate studying evolution, for example, right? So this is basically all of ecology without the part of ecology that also accounts for evolution. And the going to complex interactions and structures, sometimes the assumptions that are made are that these interactions at any level of organization are simple and unstructured. So what do I mean by this? So one example that I generally give is that if you have somebody, think of disease spread, right? This is an ecological problem, and people have studied this quite a lot from uh, simple and highly complex perspectives. So I usually like to give this example that generally a lot of classical disease models, we've moved a lot beyond that now. What they assume is that that there's a well-mixed system, right? All hosts and all parasites and all infected and susceptible individuals, all of them can interact with each other in some way. But in fact, this is kind of trivially not the case, right? So for example, I'm on Zoom right now. So if I'm super sick and carrying a lot of parasites with me, this is not going to affect any of you because you're all in different parts of the world and different spatial locations. So this means that there's some kind of a structure to interactions between organisms, right? You can only interact with those organisms that are locally around you. That's one example, but this also holds true for different at different levels of organization as well. And finally, when it comes to spatial and temporal heterogeneity, one assumption that's often made in the case of time scales is that ecology is faster than evolution. This is made both in just like ecology. When you do ecology, you kind of implicitly make this assumption. But also within the field of evolutionary biology, this assumption is often used. There are obviously complications where the relative timescales of ecological and evolutionary processes and patterns may not actually be what we want our models to do. What we want our models to kind of capture, they might actually be quite different. So you might have ecology on the same timescale as evolution in certain certain phenomena. And finally, more on space later, because that's what we are going to be discussing in the results. Okay, so with all of those assumptions in mind, one way in which we can try to start relaxing these assumptions and kind of investigate what are the consequences of relaxing these assumptions um, on trying to understand ecology, evolution, and feedbacks between ecology and evolution is that of networks, right? So what are networks? Generally, we have these entities like molecules, individuals, species, or spatial locations that are represented by nodes. And this kind of captures pairwise interactions because if two nodes are connected, then this means that there is some kind of an interaction between these two these two nodes, right? So we can take an example at the level of molecules. So you can imagine if these are two genes, and sorry, these are two genes um, within a genome, right? So I'm not talking about genes of different organisms. These are two genes within a genome, and they they kind of produce these transcripts which regulate each other's expression levels, right? So you can call these gene regulatory networks, and so you can get pairwise interactions and have different strengths of interactions, which are specified by these edges. And obviously, you can also do this, embed these networks within networks. So if you have an interaction of genes, you can have embed these gene regulatory networks inside individuals, and have inter-individual interaction networks within these populations, and then species interaction networks, and so on and so forth. So you can actually have this hierarchy of networks which can be used to model hierarchical organization. Finally, you can also, one thing that obviously we can do with networks is also specify local dynamics. So you can write down a set of equations which capture the dynamics of how these nodes change in time and how they influence each other's behavior. And finally, you can also model spatial networks, which I have here kind of as spatial locations. Okay, so with this modeling framework in mind, I would like to sort of bring up my first research theme, and this is the main thing that I'm going to discuss today, even though I'm interested in other things, which I'm going to just quickly show you at the end. So my first research theme that I'm interested in is how do biological systems properties, so scales of organization, structures in structured interactions, spatial temporal heterogeneity, impact ecology, evolution, and ecoevolution. And so the kind of scheme that I'm using currently to study this problem is as follows. So we start from a simplifying assumption, right? Uh, I, and then we kind of take this simplifying assumption and come up with a biologically inspired network representation. So I'm going to get into what this means when I present my first research piece. And the second, and then using this biologically inspired network representation, we we investigate properties, mechanisms, and patterns in ecology and evolution and their feedbacks. And the first example that I'm going to talk about is that of host-parasite ecoevolution in spatial networks, and that's all I'm going to talk about today, because I thought that I could get into some of the details to try to explain how exactly I'm kind of approaching this problem. Okay, so this is the published work, and these are the affiliations and the funding agencies that I was associated with when I did this. I did this during my PhD in Montpellier in ISEM. And so the first question is that why does space matter, right? So if you imagine these different habitat locations, so you just imagine that there are these different habitat locations on Earth, and they obviously so what happens is that, as I said, space determines what organisms interact with what other organisms, right? And also kind of the local abiotic environment, because if you're in a specific location in space, this determines what exactly you're experiencing in terms of pH, and by you I mean the organism, but what the organism is experiencing in terms of its abiotic environment, but also in terms of its biotic environment. So what other organisms it kind of interacts with. And this obviously impacts birth, death, and dispersal processes, as I mentioned, which can further change the abundance and distribution of organisms. But also in evolutionary biology, the mechanisms of gene flow very clearly depends on space, right? So what organisms enter a particular habitat patch and what organisms leave a particular habitat patch depends on space. Also, natural selection, genetic drift also kind of matter on this, because if you have these local abiotic and biotic environments changing, this as a result of space or spatial structuring, this obviously changes natural selection, because this would feedback on differential birth, death, and dispersal rates. So space matters for both ecology and evolution in this way from a very mechanistic perspective. And so generally when people model spatial structure, people make a lot of different assumptions, though obviously, again, as a disclaimer, not all studies do this. But there are some classical assumptions that people make, right? So one of the classical assumptions is that you can kind of ignore the local dynamics. So you can kind of say that we don't really care about these birth, death, dispersal processes of these organisms, but we rather model these patches, these habitat patches, as a population itself. And that's called the Levin-style metapopulation model. And that was a super useful model to try to actually start understanding spatially structured systems. You can also explicitly model local dynamics, but make other simplifying assumptions. So one of the simplifying assumptions is that that dispersal is possible between all of these patches. So you can imagine an organism right here where I'm pointing to, and this organism disperses, so it kind of moves from one patch to the other, and it can end up at any one of these different habitat patches. So that's the kind of assumption known as the island model assumption. And not only is this like a and many times for analytical simplicity, people assume that there are like infinite number of islands. There are some more spatially structured systems that people consider, stuff like regular grids or lattice models. So there's like a whole set of different assumptions people usually make, assumptions or modeling choices people make when studying spatial systems. So now what happens when you start thinking about spatially structured systems from a network's perspective? You go from something like this to something that looks like this, right? So these are some kind of river landscapes. This is like rivers, and these are terrestrial landscapes. So this is just to say that you have these two kind of canonical ecosystems, riverine ecosystems and terrestrial ecosystems. And if you try to look at these island model approaches, it's obviously not going to be the case that an organism can disperse from here all the way to here just like that. Well, it is possible, but generally if it's just moving along the river, it's not going to be dispersing from here all the way to here or here all the way to here, right? And to capture this kind of spatial structure in terrestrial and riverine aquatic landscapes, we can actually represent them using spatial networks. And so let's start by talking about the river network first. So you can imagine a river structure, right? There are a lot of different branches, and then you have these upstream things that kind of go and converge into an outlet. So one feature of these river networks is that they are heterogeneous. So you have a lot of habitat locations or patches, as I said, which have only one other neighbor, because there are a lot of branches, right? So this has only one other neighbor. The one that I'm pointing to, it has only one other neighbor. This guy also has one other neighbor, one other neighbor. And there are very few patches like this one which have more than one neighbor. So this has three neighbors, this has a lot more neighbors. And this kind of thing we call as heterogeneity in degree distribution. Degree is the number of neighbors. And there are certain more properties that these networks can capture, and these are known as optimal channel networks. They come from the earth sciences, and they've been studied quite extensively in ecology as well, to try to understand, for example, the diversity of species in river systems versus diversity of species in non-river systems. The other type of landscape that we can think of, as I said, are terrestrial landscapes. And to represent this kind of terrestrial landscape or to capture their properties, we can imagine that there's a square piece of land like this. So from a uniform 0, 1, 0, 1 distribution, there are these habitat patches which are drawn from this kind of a distribution. So you can imagine uh, 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 just a square piece of land with uniformly distributed habitat patches. And obviously, again, we want to capture some kind of structure. So we say that that organisms can disperse only locally, so they can't disperse long distances. So we kind of cluster these patches by the fact that if they're within a small radius, they're connected to each other, and if they're outside the small radius, they're not connected to each other. And this forms a structure known as random geometric graphs. And this can be used to represent terrestrial networks, because this can capture these kind of modular structures which are a feature of terrestrial networks. And these are our biologically inspired network representations for spatial systems, so riverine and terrestrial biomes. So we've gone from kind of more simplified systems where you can have dispersal from anywhere to kind of a structured system inspired by kind of what we think is going on in biology. And obviously, the next part is to kind of try to understand why we are focusing on host parasite systems. So this is because host parasite systems are everywhere. They're found in all of these different ecosystems. And they are quite interesting from both a fundamental and applied perspective, because host parasite systems in ecology potentially regulate population densities, and in evolution there are these strong interactions between hosts and parasites, which are quite interesting. So this is kind of our model system for today. And to try to understand how spatial network structure influences host parasite systems, we built an individual based model of host parasite ecoevolution. And we focus on a very key trait of parasites, which is that of virulence, right? So virulence is the extent to which a parasite harms its host. Uh, we have, and we are going to study the evolution of virulence. We assume that increased virulence is a byproduct of increasing transmission. So what this means is that parasites need to exploit their hosts in order to be able to reproduce more. So this implies that any harm that the host causes to any harm that the parasite causes the host is a byproduct of this increasing transmission ability. So what you need to remember is just that there's an increasing relationship between virulence and transmission. We allow for the parasite to evolve for a long period of time to try to understand its behavior. And so this is an individual based model. So what we do is we specify these network structures, and then within a patch in this network we model individuals. These individuals can be either susceptible if they don't have a parasite, or infected if they have a parasite, and the kind of parasite trait is stored inside the parasite, and this can evolve with time. So we introduce variation and mutation in these traits. And again, this is a simulation model, and this is but it's kind of a relatively simplified simulation model. So I'm going to go into the life cycle of what this simulation model looks like. I'm just going to check the time to see. Yeah, I think that's fine. I can go into this. So this is the life cycle that we model. So we have these different individuals on a patch, right? So this is a patch. These are the individuals. This is a susceptible individual. This is an infected individual. After organisms are born, they can disperse. They disperse with a probability d from one patch to the other, and the hosts carry their parasite when they disperse from one patch to the other. That's an assumption. Then after this, hosts in the target patch, they reproduce. They produce a number of offspring given by this Beverton-Holt model. And susceptible individuals produce more offspring, infected individuals produce fewer offspring, and this reduction in the offspring is given by the virulence, right? So virulence acts on the fecundity of the host. So infected individuals produce 1 minus v times offspring as susceptibles. Going from here, after reproduction, there's parasite transmission. This is generally after the parental generation dies, they release these parasite propagules, which can infect the offspring generation. And during this process, we assume that one mutant sort of so you can see these slightly different colors in pink and red. So these are different mutants, different types with different virulence, and their transmission ability is also genetically encoded via virulence. So the more virulent the parasite is, so this the more it can infect, the more number of hosts it can infect. And then this kind of goes on, and we iterate this for a long period of time to get an evolutionarily stable or a long-term value of virulence. And so let's now get into the first result over here. So when we look at these terrestrial and river networks, how do we expect parasite virulence to evolve as a function of host dispersal? And what we find is, in fact, that the river networks and terrestrial networks produce distinct patterns of parasite virulence with host dispersal. And host dispersal is on the x-axis because you can imagine that if dispersal is very low, these patches are essentially uncoupled, right? But as dispersal increases, patches become more coupled, so the spatial structure becomes more apparent and more important. So here we see that virulence is very similar because essentially these systems behave very similarly. Since these patches are mostly uncoupled. But as dispersal increases, the differences between these networks also increases or changes. And in terrestrial systems, we get this kind of saturating increase of virulence with host dispersal, whereas in riverine aquatic systems, we have this kind of a unimodal shape. So we get distinct patterns of parasite virulence in terrestrial versus riverine spatial networks. So that's the first result to remember, right? So evolutionary patterns are impacted by spatial network structure. So the next thing is that, well, we could just stop here and be happy, but we should not be happy to stop here, because we would like to try to understand why this happens and how this happens, right? So we are also interested in processes and mechanisms. And I'm going to give a small summary of what the literature says. So this is what work has been done before. And what this literature says is that virulence evolution in spatially structured systems is driven by kin selection. And I'm going to try to quickly explain this. I hope this is clear. We can maybe discuss this later if this is not super clear. So the first thing that I mentioned again is that greater virulence implies greater transmission. And in the case of fecundity virulence, this specific type of virulence that I've assumed in this model, in a non-spatial system, so in a well-mixed system where all hosts and all parasites can interact with each other, we expect virulence to be maximized. And the reason for this is because if you have more virulent parasite, it can transmit more. So it maximizes the number of hosts that it can transmit to. Which leads to the evolution of maximal virulence. But in spatial systems, what we have is there's differential clustering of related parasite genotypes, right? So if you have very low dispersal, for example, you would expect a lot of very related individuals, related parasite individuals, to be clustered in the same patch. And because of this, lower virulence may evolve. So lower virulence relative to maximal virulence would evolve as an altruistic strategy. So this means that there's a cost to evolving this. There's an individual cost to evolving this strategy, but an inclusive fitness. So there's a benefit to related individuals of evolving this lower virulence strategy, because this allows for a larger pool of uninfected hosts for kin. And just to note here that this is assuming that host densities are kind of comparable across these scenarios. But in fact, if host densities are very different, then this can also change things. So the main thing I want you to remember here is that if you have highly clustered and related parasites, this allows for lower virulence to come up, because this allows for a larger pool of susceptible hosts for related and kin related individuals. And so we can actually test this in our simulation model, because what we have here is by default there's some kin structure, right? When there's lower dispersal, we have a lot of clustering of kin. When there's higher dispersal, we have less clustering of kin, and so on and so forth. So we can actually break this kin structure in our model artificially by shuffling genotypes around. And what we find when we do that is that we get this maximal virulence. And if you remember from the previous slide, I said that this is what we expect in the case that the system is well mixed. So this is not exactly a well mixed system, but we are actually breaking kin structure. So this is what we expect in the absence of kin structure. We expect maximal virulence in both networks, and this is what we get. So this is the first line of evidence that this is that this is what is happening in our model. We have a second line of evidence, which is that we can actually measure relatedness in parasites in these models, right? So we can actually mark these parasites with a neutral locus and measure how related they are to within a given patch. And when we measure this relatedness within a patch as a function of host dispersal, so this is relatedness on the y-axis and host dispersal in the x-axis, what we see is that when dispersal is very low, relatedness is very high, implying lower virulence, which is perfectly in line with what I just said, where we expect lower virulence when parasites are superclustered. Then in riverine networks, we have lower relatedness relative to terrestrial networks here at intermediate dispersal, leading to this peak. And as dispersal increases again, relatedness increases again in riverine aquatic landscapes, so or in river systems. So what we see here is that virulence is a mirror image of parasite relatedness, which is another kind of evidence that kin selection is what drives evolution. And the second thing to note here is that these networks produce characteristic patterns which drive parasite virulence evolution. So we can actually, if I stopped here, then I would just like be able to apply this intuition to this specific system, but we would actually try to we really want to understand what these spatial networks are doing, right? So I'm going to focus on the river system and try to explain why we actually get the patterns of parasite diversity or parasite relatedness. Uh, so just to say that the more parasite diversity, lower relatedness, just to say that. Um, so why do we get that in river networks? And this comes about from this heterogeneous structure, right? So I mentioned very early on that these river networks have a highly heterogeneous structure. So this means that there are many patches like this, the ones that I'm pointing to here, which have only degree 1, and there are few patches which have a very high degree. So when dispersal is low, right? When dispersal is low, we have so many of these degree 1 patches, um, because of which there are these parasite diversity is low, because you don't have too much incoming variation from different patches, because they're quite decoupled from each other when dispersal is low, and they're especially more decoupled even than terrestrial landscapes because of this low connectivity. But what happens when dispersal increases, right? We see that these river systems, in spite of their lower connectivity, actually lead to higher parasite diversity. So what happens is that when dispersal increases, right, this increases the asymmetry in parasite densities. So if we just measure the number of infected individuals, you would see that there are a lot more infected individuals in these high degree patches and very few in these lower degree patches. And an intuitive explanation for this is that these high degree patches, they receive so much from all of these other patches, right? So they receive a lot, and then they receive a lot more than they send. And these low degree patches send a lot more than they receive, because they receive only from one patch, and they send but they send everything to this patch, but they receive something that's divided between so many different patches, right? So this creates these asymmetries, and obviously you can imagine that as dispersal increases, these asymmetries should increase. So at intermediate dispersal, what happens is this kind of prevents the extinction of parasite in these low degree patches, which increases diversity. So this allows for these landscapes to maintain a lot more diversity than we would expect, um, just from their connectivity. And finally, as dispersal increases again, this asymmetry increases so much so that now these patches are so small that there's not so much going into these high degree patches. And because of this, again, further increased asymmetry, parasite diversity decreases again. So that's how we get these patterns of parasite relatedness in our system. So we can actually go one step down and outline what happens exactly within this river network. Okay, but we can actually now show this, because that this is because of heterogeneous degree distribution. And by doing this, to do this, we kind of do a trick where we design these networks which have otherwise different properties, right? This looks very different to this. I call this kind of spiky network, but you can call it whatever you want. But this has a very heterogeneous degree distribution. So this also has many degree 1 patches, which you can see here, and very few patches of higher connectivity. And we have this kind of circular network, which is low connectivity, just like the river network. And what we see here is that these spiky networks recapture evolved parasite virulence as a function of host dispersal, at least qualitatively. So we get this maximal virulence evolving plus this reduction afterwards. And whereas in circular networks, we don't get this, right? So we just get this kind of saturating increase. So this again points to heterogeneous degree distribution of river networks, uh, uh, which structure density and genes, and this is what leads to differences in parasite virulence evolution. And so to sum that up, to sum up the main results that we talked about today, um, so we have our terrestrial and river networks here. What do these terrestrial and river networks do? They structure host parasite densities, which changes the local biotic and abiotic environments. This changes both natural select. This changes our evolutionary mechanisms. As in our case, this was kin selection, which drives the evolution of parasite virulence. So these networks, because of their properties, structure both population densities and genes in specific ways, which allows for evolution to proceed in certain ways, which leads to differential evolution. In the case of host parasite systems, we are looking at parasite virulence evolution. And this may also feed back on the epidemiology of the system, which I'm not going to be discussing about so much right now. And so one thing to notice is that because we kind of focused on trying to really understand what happens um, uh, quite in detail because of this heterogeneous degree distribution, some of our insights from these results may not just apply to parasite virulence evolution, right? So if we have similar or comparable ecological conditions, this should be we should be able to extend these results to actually other traits. And I'm going to just just going to mention one of our studies doing this, but we are actually this is not just about virulence evolution, because this part should not change, should not be too different between different traits, and perhaps between different systems, though this would depend exactly on the specification of the ecological dynamics and the evolutionary dynamics. And the other thing that I'm working on right now is also looking into some analytical results, not in the case of virulence evolution, but for other traits, because just to give some background, there people kind of have a very good understanding of kin selection in these infinite island models that I mentioned, where all patches can interact with all other patches. But when it comes to spatial networks, these results have not yet been generalized. So I think that we can try to understand these classical results in terms of these spatial networks, and I think that could be very interesting direction. So these are a few applications of this approach. But I also want to just quickly note that networks and eco-evo, this is not the only example, so this is all I'm presenting today, but we actually extended this study to also include host dispersal evolution, because actually this heterogeneous degree distribution that I mentioned actually strongly impacts the evolution of dispersal itself, right? And so this study, it's up on bioRxiv, so if you want to read more about this, that's right here. Plus there's some unpublished work again in this spatial network context, which I'm working on. And the other thing I want to again emphasize, that this is not just about spatial networks, right? This can be done, this approach can be used elsewhere. And so one study that I was involved in, or I actually led, was this on the genetic architecture of dispersal and local adaptation. So we kind of try to study how genotypes go to phenotypes. Usually these are very simple models, again, going into our simplifying assumptions framework, and we replace these genotype to phenotype maps with gene regulatory networks, trying to get at what happens across across different scales of organization, going all the way from genes to the range expansions, which are like an ecological phenomenon taking into account spatially structured space. Just to say that there are a lot more things that can be done. It's not just spatial systems, but actually this can be applied to different levels of organization as well, and also across levels of organization. And finally, with this, this is what we had here, right? We had our simplifying assumption, biologically inspired spatial network representation, and ecological process mechanism and patterns. So I gave you the one example in detail and some other examples in no detail at all. And while this is I still believe that this is a very good line of research, and I am personally involved in this line of research. I think we kind of need to also move beyond just looking at different networks and what are these, like, okay, you have different networks, they give different behavior, this is really cool, this is awesome. But I think that there's a second related and complementary question, and that's what I'm working on right now. So in case you want to discuss this with me, or if this is just to kind of stimulate more discussion, or if you want to talk to me about this or collaborate potentially, this is the kind of direction that I'm going in. So that's my research theme 2. And this research theme 2 basically is, how do simple ecological and evolutionary patterns emerge from complex networks? So in this, we kind of start from the reverse direction. We actually start from ecological and evolutionary patterns, or mixed. So currently I'm just looking at ecology and evolution separately, which emerge in theoretical or empirical studies. So one of them that I'm studying currently right now is that of fitness effects of mutations. So you can imagine that if you have different mutations which change fitness in a certain way, indicated by delta X, but this could be any phenotype, and you have the same mutation here. So there's a very low dimensional relationship between this fitness effect and the fitness of the strain to begin with. Usually very simple linear relationships. So when we have these kind of ecological and evolutionary patterns which are very simple, then one can also ask that how do these patterns really emerge in spite of the fact that there's underlying network complexity. And there are many different approaches people use, many times coming from statistical mechanics, uh, to try to understand how these simple patterns emerge. But I'm going to try to understand them in terms of these complex networks. So that's my kind of future direction slide, which is not very clear, but if you want to talk to me about this, I'm always happy to chat. And finally, this is what I'm working on during my postdoc in the Krasnymsky lab in UC San Diego. So if you want to chat to me about this, this is also something that I'm interested in. Yeah, that's about it. Thank you. Sorry if it was a little unclear towards the end. And I'm happy to take questions.
That was quite clear to me, at least. So I think you did great. Thanks for the talk. It was quite nice. So yeah, as always, everyone raise your hands and I'll sort of go through one by one, whoever's on the thing.